Hi, this is Ray, and I'd like to share you about our recent work, exploration of dimension reduction techniques for clustering spatial transcriptomics data. I'd like to begin with a metaphor. The bulk RNA sequencing is like a fruit smoothie where everything is mixed. Single-cell RNA sequencing is like tasting each individual fruit, while spatial transcriptomics, ST, provides a fruit platter view, preserving the spatial context. Spatial domain identification, SDI, is a critical task in ST, allowing us to gain deeper insights into the tissue microenvironments and biological functions. Clustering is a necessary step in SDI, serving as a general method to assign clusters to each spatial spot. And here, we focus on enhancing this clustering aspect. We present a dimensional reduction method using Giotto and model-based clustering that eliminates the need to predefine the number of clusters or model type, achieving more promising result than principal component analysis. In a publicly available reference dataset, DLPFC, dorsolateral prefrontal cortex. Both statistical and deep learning models have been developed for SDI. Giotto as a statistical model uses a hidden Markov random field to detect spatial domains with consistently gene expression patterns. Meanwhile, deep learning models such as GraphST employ graph convolutional neural networks to reconstruct the gene expression profile with the spatial context. However, most SDI methods use PCA for dimension reduction, has a fixed number of clusters. In practice, the PCA approach might not capture the nuances needed. Also, the ground truth annotations, like the actual number of clusters, they are generally unavailable for most of the dataset. To address those limitations, we applied model-based clustering with various dimension reduction techniques. Specifically, we optimized the clustering process in the following two aspects. First, we used the Bayesian information criterion to determine the number of clusters and model type. So the Bayesian information criterion BIC is a statistical criterion that balances the model fit with complexity. For example, the default MAMCLUS model used by GraphST is hard-coded as model EE, and the number of clusters is also hard-coded as 7. The model EE stands for ellipsoidal equal volume, equal shape, and equal orientation. But anyway, in general, the big value is encouraging to stack simple models as big are used to reduce the risk of overfitting. By using big, we don't have to hard code the model and the number cluster anymore. Second, we apply the SVG-based dimension reduction method instead of PCA to reduce the dimensionality of the spatially enhanced gene expression data generated by GraphST. Specifically, we use Giotto to identify genes with distinct spatial patterns, which we call SVG, the spatially variable genes. We then selected multiple numbers of top SVGs and test the big value to help us to determine the optimal number to select. Below, you can see in the chart, is an example of how we use the ALBO method with BIG to select the optimal SVGs for sample 15, 16, 7, 3. In this example, we select 65 as the optimal number with a pretty good adjusted rank index, ARI. So the ARI is a metric to evaluate the clustering result. It ranges between -1 to 1, where -1 means completely different clustering assignment and 1 being totally identical. While this ALBO method may not yield the highest ARI, but it's sufficient as we don't have the ground truth information during clustering, thus we cannot use the ARI as a evaluation metrics for determine the number of SVG. So we evaluate the clustering performance by calculating the ARI, by comparing the result clusters with the DLPSC ground truth. Our experiments are reproducible with a fixed random seed. We also tested a range of random seed, and our approach consistently outperforms GraphST's PCA on 8 out of the 12 samples in the reference dataset, as you can see on the results section. And for sample 151673, as you can see, it reached an ARI of 0.6198, which is higher than the PCA default. Our method has some limitation though. One limitation that we encounter is that the album method sometimes fails on certain datasets. This might indicate the needs for spatial clustering metrics to help determine the best number of SVGs. We're also working on our generalizability and the scalability of our method. So if you have any questions, please do not hesitate to ask, and I'm happy to answer those. And thank you for listening.